Hello again, everyone. Welcome to part 2B. So this is the fourth installment in this week's material. This is for session number 2. So we're going to finish up here about 25 slides, and then we'll have completed the lecture material for this week. And just a quick reminder that with this picture right here, what you should be able to do is have a sense. Now, you might know the exact answer for where this is, or you might come up with 2 or 3 possible answers. That's just fine. The thing here is to make sure that you have a familiarity with this area where 1 is, where 2 is, and 3, and so forth. Rather than worrying about an exact answer, you say, what are the possible answers in that area? Because I promise you, if I'm going to use a picture, I'm going to try to be as specific as I possibly can. So use this as review and understand that there may be more than one correct answer, depending on where you see that pointer actually locating. And now we're going to move into the accessory organs. The 3 primary accessory organs that we plan to talk about are the pancreas, the exocrine pancreas primarily, though there is an endocrine function as well, the liver, which has a complex cellular architecture, along with the accessory organ that helps store some of what's called bile, called the gallbladder. Now, the accessory organs, the liver and the pancreas specifically, are what we have what we call exocrine function. That is to say, you have cells that are secreting into a dedicated duct that then leads to the external environment. In this case, the external environment is the lumen of the digestive tract. Now, the gallbladder is not really a gland per se. It does store bile, and it releases it. But you can actually survive without the gallbladder. It's not ideal, but it's not a problem if you have to have it removed for certain reasons. But both the liver and the pancreas are exocrine glands, at least in terms of the digestion. So the pancreas is about the size of your hand if you sort of roll your fingers together. It's got a pronounced head. It's got this tail, if you will, and it has both exocrine and endocrine capabilities. Exocrine secretes digestive enzymes along with a material called bicarbonate, which helps neutralize the stomach acid and raises the pH of the chyme up to around a pH of 5 or 6. Still a little bit acidic, but not nearly as acidic as the stomach acid. It has these ducts, so you have the pancreatic ducts that then feed through a sphincter, into the duodenum, or the base of the duodenum at the beginning of the jejunum. Again, with the exocrine function, we're secreting enzymes and bicarbonate. The cells that secrete the enzymes are known as acinar cells. These are exocrine cells, and they're releasing the majority of the enzymes that we see in the digestive tract. With the exception of some enzymes in the stomach and in the saliva, almost all the rest of them are coming from these acinar cells. So we have and we've seen these enzymes before. We have, fat digesting enzymes, lipases, protein digesting enzymes, peptidases. That includes trypsin, chymotrypsin, and an enzyme called elastase. We also have a carbohydrate. This will break down starches and complex carbohydrates into shorter sugars, sometimes 3, 4, 5, maybe as few as 1. Then we have nucleases that break down RNA and DNA, and then these guys here are enzymes that will recognize double sugars like lactose and sucrose and maltose and break those into simple sugars like glucose. And we have that bicarbonate. Okay, this is going to reduce the acidity, so we're going to bring the pH up to more like a 5, 6 range, if you will. And so we're neutralizing the acidic chyme into, again, a higher pH, which allows these enzymes up here to function properly. And we also have an endocrine side. The 2 most common and well-known hormones from the endocrine, these are called the islets of Langerhans. They are pancreatic islets. Um, what an endocrine system is, just to remind you, is that you're secreting directly into extracellular fluid. And you're not going into the blood plasma yet. You have to go through the extracellular fluid first, and then they can migrate into the blood plasma. But insulin and glucagon. Insulin causes glucose uptake by muscles and fat and other tissues. Glucagon causes release of glucose from the liver, and from fat and storage cells in the body. And this is not the best picture I've ever seen, but these purplish cells, the darker purple cells with the light interior, those are the acinar cells. And you see these little crack looking things, these little crevices, those are part of the pancreatic duct. They're very, very small here, and they collect and they get bigger and bigger as you get closer and closer to the head. And these lighter areas, these are the islets of Langerhans. All right, so these are the pancreatic islets. These are the endocrine uh, uh, tissues in the pancreas. And we have three here. We have alpha, beta, and delta. There are actually about 5 or 6 cells, each secreting different hormones. The alpha cells are the glucagon, and the beta cells are the insulin. Those are two huge ones. Then this last one here, this is an enzyme called somatostatin, which is going to slow down the digestive function of the stomach. And those are secreted by what are called delta cells. So just a word or two about glucagon and insulin. Glucagon causes release of glucose into the bloodstream, primarily by breaking down the polymer of glucose called glycogen. So this process is glycogenolysis. This will promote glucose production inside muscles. Muscles generally won't release glucose, but the liver does. So in the liver also stores glycogen. You usually, give or take, when you're fully rested, you have about a pound of glycogen in your body, some of which is in the muscle, some of which is in the liver. And in the muscle, you'll break down the glycogen, but that's in order to serve the muscle. But the liver will also break down glycogen and release it into the bloodstream, and that's mediated often by this hormone glucagon. Then you have insulin. Everybody knows about insulin for diabetes. This triggers glucose uptake. This can be into fat cells to create fat, or in muscle cells and in liver, because again, the liver will want to store glucose as glycogen. Muscle cells will use it to make ATP, or if they are stocked up on ATP, they'll also store it as glycogen. Now, the brain and active muscle, and especially heart muscle, don't need insulin to take up glucose. In fact, if you administer insulin at the wrong time, and when you don't have adequate glucose in your bloodstream, that can actually cause your heart and your brain to go into crisis. We call that insulin shock, because there's not enough, because they take up glucose all the time. Neural cells and heart muscle cells need glucose all the time. They can't go without it. So they're always taking up glucose. But your skeletal muscle, there's enough of it. It's about 40 or 45% of your body mass. So you really only want to take up glucose in the skeletal muscle and the liver when you have lots of glucose in your blood, and that's when insulin kicks in. Now, I'm not entirely sure why they included thyroxine in here, but it's probably due to the fact that this will modulate glucagon and insulin indirectly. So it's one of these sort of more controlling hormones. Yes, it controls basal metabolic rate, but hormones do interact with each other, and thyroxine, if you have more of it, will increase metabolic rate, which means increase glucagon and suppression of insulin. Some basic anatomy of the liver. An interesting factoid, there are actually 4 lobes. So we have a right and a left here, and then there are some in the back that we can't see in this picture. But there are 4 lobes, which in each lobe is able to regenerate on its own. So if you have an organ donor, you can take the liver from that donor and donate up to 4 lobes to 4 different people, and they will regenerate in a way that will allow for liver function. So it's one of these really, really interesting organs that has four lobes, each of which is sort of autonomously regenerative. Now, again, we have two of the four lobes here. There's a central sort of ligament called the falciform ligament. You can see the gallbladder kind of tucked in under here. It's not exactly a healthy color. It's usually a little more of an olive drab color, but there it is. Um, not much else about the liver. It's fairly homogeneous when it comes to macro anatomy. And so here's another lobe here. This is the caudate lobe. This is looking from the back. And then there's the quadrate lobe. That's the fourth lobe in there. Here you can also see, again, it's hard to see, but there's a lesser omentum that's hiding in here. Again, here, better view of the gallbladder, okay, and the bile duct. You can see a hepatic vein, or you can at least try. This is not a great picture, but again, just some basic, lobe major structure. Again, here's the ligament, the round ligament that's part of the suspensory structure involved in the mesenteries and the omenta. Where the real action is in the liver is in these lobules. You can see it has a roughly pentagonal or a hexagonal, and if you were to take a histology course, I guarantee you by at least the first quarter of the semester, you could spot liver from a mile away. It has a very unique cellular architecture, sort of like bone. You have this central vein, going on, and then there are some other ducts that are not readily visible here, but it has this really interesting, architecture that really is sort of serving as a quote unquote biochemical filter. So we've got the portal vein, you've got the bile duct, and you've got the hepatic artery. We call that the portal triad. Again, the liver has a sort of, it's not a true filter, but it is a sort of biochemical filter, and it's actually metabolizing lots of organic compounds as blood moves through that portal vein, and then eventually it's distributed back into the blood. So we have some functions we want to go through here, and I'd love to spend like 4 or 5 minutes on this slide. I don't think I want to spend that much time, but do at least be able to recognize these as potential functions of the liver. Since we have that hepatic portal vein, the liver is one of the front line areas for detoxifying potential toxic compounds. This includes drugs. Part of the reason why we have to take a larger dose when we take things orally is what we call the first pass effect, where the liver will actually remove those drugs before it ever gets to the rest of the body. The liver handles fats, the liver handles carbohydrates, the liver does it all. It's one of the most biochemically intense tissues in the body. And so we can store excess nutrients there. And that's why liver is one of the most nutritious foods if you're going to eat meat. Now, in our Western diet, liver doesn't seem to hold the appeal that it might have had 100 years or so ago. But nevertheless, liver is much more nutritious than muscle, which is what we normally eat in steaks and so forth, because it has lots of vitamins. It certainly has a lot of vitamin A. In fact, polar bears, their livers have so much vitamin A that if you were to eat polar bear liver, you'd actually suffer vitamin A toxicity and die. People have died from eating polar bear liver. A, D, E, and K, the fat-soluble vitamins, huge sink for iron. In times of increased need, like in pregnancy, or if you happen to have an iron deficient diet, there is iron stored in the liver in order to allow for proper manufacture of hemoglobin. Then we have other metabolic functions. Certainly we have a digestive function, bile salts, and a big component of bile is a metabolite of hemoglobin called bilirubin. So when a red blood cell ages out, and that's at about 4 months, the blood cell's broken down, and you don't want that iron just floating around. That iron is nasty reactive. In fact, the reason we use iron is in order to solubilize oxygen. Oxygen's not very soluble, and so we use iron in hemoglobin in order to make the oxygen more soluble in the blood. But that iron is incredibly reactive, and we have to keep it caged. So when we break down a red blood cell, we metabolize that hemoglobin into bilirubin. Some of that bilirubin escapes into your urine and gives you your urine its yellowish color. But also the brown in feces is actually the brown of bilirubin. The majority of bilirubin is removed through the fecal route, through the bile. The liver also manages cholesterol. It can synthesize cholesterol if you don't have enough in your diet. You don't ever need to eat one molecule of cholesterol in your body. Your liver will make it if you don't have it in your diet. But because we tend to eat relatively high-fat diets, the cholesterol kind of comes along for the ride. But the liver can actually manufacture cholesterol from fatty acids. The liver also makes the major plasma proteins, including one called albumin, which is a major carrier protein for steroid hormones and drugs. Like when you take certain drugs, many of them will distribute in the bloodstream inside of albumin. Then we have clotting factor here. This is thrombin is the major clotting factor, and the precursor of thrombin is prothrombin. Liver handles fats, so it will help make lipoproteins to help manage fats in the bloodstream in and out of the fat tissue or other cells in the body that have need for like di and monoglycerides. It handles nitrogenous waste, so we can convert amines into urea. We can also create glucose from amino acids, so gluconeogenesis. We can synthesize glycogen and store it inside the liver, or in times of fasting, we can break that glycogen down and release glucose into the bloodstream. There are certain disorders where you can make glycogen, but you can't break it down, and that's a real problem because you would suffer from very, very low blood sugars because you couldn't break down the glycogen in order to sustain at least a normal resting blood sugar. And like I said, handling fats, storing fats is also part of it. In fact, there are certain diseases called fatty liver disease. And that's what we're going to talk about here. We've got fatty liver disease. This usually occurs because of some injury, chronic alcohol intake, poisonous mushroom intake, drug use, intravenous use, you name it. Because you know why? Because the liver's working overtime to try to detoxify those drugs. Cirrhosis is just an advanced form of this. This is where you have outright scarification of the liver, and now you've completely lost function in that tissue. Hepatitis is usually a viral disorder. That's inflammation of the liver. And during inflammation, you generally have loss of function. So what do you see when somebody has hepatitis? You typically see jaundice because their bilirubin builds up. Or they have they get swelling and they get ascites in their belly because they're not making the proper plasma proteins. All sorts of things like that. I've had not one but two friends basically die of chronic alcoholism, and I sort of watched them deteriorate. It was heartbreaking because we were trying to intervene, but they just didn't respond to that. But now the liver can regenerate itself. You only need about 1/10 of your liver in order to fully regenerate a new liver. That's why liver transplants are more common than other transplants because you can donate usually up to 4, maybe 5 recipients for any one liver. You don't have liver function though, you're in deep trouble. And like I said, one of my friends, I saw him just a couple of weeks before he passed. He had looked like he had aged. He looked 20 years older than I thought he should have looked. It was actually quite stunning, and unfortunately, he passed. Now, this slide looks complicated, but there's really only one structure that we're really interested in, and that's this hepatic portal vein. All these other veins that are coming off all these different organs, the stomach, the spleen, pancreas, small and large intestine, through all of these blue structures here, these veins are all feeding into the hepatic portal vein. Roughly a major percentage of blood that comes out of the digestive tract runs into the liver and then gets processed by the liver. It's like I said, a biochemical filter, then comes out and leads into the inferior vena cava and back to the heart. As we mentioned earlier, you can live without your gallbladder because the gallbladder is generally a storage structure storing what we call bile. It has about the same consistency as coffee or tea, bile does. 95% water. It has ions in it. Some of them are rather complex. It has small, short peptides. Certainly has cholesterol in there. Lots of bilirubin, which is metabolized hemoglobin. Sodium, potassium chloride. That's just because they're basically everywhere in the extracellular fluid. A few lipids and some proteins, and this mixture acts as a sort of surfactant. Now, what does a surfactant means? It means that it breaks down the surface tension of liquids. In this case, it will break down the surface tension of fatty droplets. All right, so if you have a bottle of vinaigrette dressing, you shake it, and you'll see the droplets of oil in there. It does not take a lot of energy to form large droplets, macro-sized droplets. Well, we can't absorb that. And so what you have to do is you have to break up those droplets into small enough droplets that can be internalized into the lacteals. And so what we're going to do is we're going to emulsify these things, break down that surface tension. We're going to expose those droplets to lipases to break down the triglycerides into fatty acids and glycerol. Almost said glycogen. And so to make these molecules smaller and to make them absorbable. And so you can see the bile duct here. You can see the gallbladder off to the side here. It actually has some smooth muscle in there that can contract during periods of digestion to release more bile salts and bile into the small intestine. Certainly have the liver here, the tail of it. The head is kind of tucked in here with the pancreatic ducts. There's a major and a minor there. That then feed into the duodenum, neutralizing stomach acid, facilitating the activation of digestive enzymes, moving on into the jejunum. As I mentioned previously, the digestive tract is an intensely endocrine system. Intensely. And so we have three key hormones we want to talk about. Now, we've talked about some other hormones, so make sure you keep these things straight. We've got gastrin, cholecystokinin, that's a mouthful, CCK, and secretin. So we're going to start with gastrin first. It's released by the endocrine cells in the stomach and activates the stomach. All right. So if we have lots of protein, we're going to get a lot of gastrin, and we're going to increase the enzyme secretion, increase acid secretion in the stomach, because those two things, the peptin peptidases will help break down that high level of protein. We're going to churn more, certainly get the body ready for increased glucose. So we're going to start increasing insulin and go from there. All right, so it's increasing stomach activity is what a gastrin is doing, especially with protein. So we get this gastroileal reflex. We're increasing peristalsis. We are increasing motility throughout the entire GI tract. And that's one of the reasons why people frequently have to go to the bathroom not long after a meal, because of the gastrin hormone is increasing overall GI activity. The gastroileal reflex causes increase in large intestine function, and the gastrocolic reflex triggers the desire to have a bowel movement. The other two hormones, secretin and cholecystokinin, are known as enterogastrones because they affect the intestines and the stomach. Well, we've turned on the stomach with gastrins, and we're releasing this gastrin, which is causing motility and things. Well, we have to have off switches as well. And so secretin, which is released from the duodenum, will, as they sense hydrochloric acid, will turn off that gastrin release. It'll also stimulate bicarbonate release of the pancreatic juice from the pancreas. This will neutralize the chyme that you're sensing coming from the stomach and make it more neutral, anywhere from around 5.5 up to a truly neutral pH in the 7s. Cholecystokinin is when we sense fats and protein in the duodenum. Again, that'll inhibit gastrin, stimulate gallbladder contraction to release bile, certainly more pancreatic juice, as well as releasing the sphincter to allow bile and pancreatic juice to enter the duodenum. You don't want it going in there all the time. You only want it going in when there's need. So it senses so the secretin is sensed from the stomach acid coming in. The cholecystokinin is released when fat and protein are sensed in the duodenum. So some basic gross anatomy of the large intestine. There are sort of four major regions, and let's get the largest function out of the way. That the most preeminent function of the colon is as a desiccating organ. It is reabsorbing water from the secretions and food that have entered the GI tract. It's a water retention organ. In fact, if material stays in the colon for too long, it tends to compact and desiccate, leading to constipation or other types of syndromes associated with buildup of fecal material in the large intestine. So we have the ascending colon, we have the transverse colon, we have the descending colon, and then we have the S-sigmoid colon leading to the rectum and then the anal sphincter. There are other structures in here, but for now, let's focus primarily on the four major regions. So as we said in the previous slide, water absorption is the big thing here. Big time water conservation organ. You will also, as you're absorbing water, bring along a few minerals and ions. That just happens as a process of water movement. You do get some vitamins that are made by commensal bacteria here. Vitamin K, a form of vitamin K can be made in here. Not a lot, but mostly vitamin K is the number one thing. Again, here's ascending, transverse, descending, and then sigmoid. Now, below the sigmoid colon, you have the cecum, you have the rectum, and the anus. So the rectum is kind of a storage chamber. It's also a bit of a sensory organ. If you can, that organ can actually sense if it's gas or if it's solid material, etc., etc. So at the very end, you have a storage and desiccating area called the rectum, and then the removal area known as the anus. It's a double sphincter setup designed to not only store but also assess the quality of the material, if you will. So that last 20 centimeters or so. Now you can store material for hours or even days in the case of certain situations. During that time, what's happening is you're continuing to reabsorb water, compact the material so it gets denser and denser the longer it stays there. So there is a sort of window. If it doesn't stay there very long, we call it diarrhea. If it stays there too long, it leads to a condition known as constipation or fecal retention. So we have this internal and external sphincter. That stretch is what you perceive as need to go to the bathroom. And depending on the type of stretch that you get, it can detect gas or it can detect solid material. And that stretch will lead to increased peristaltic wave motion, especially in the sigmoid and rectal areas. Parasympathetic, that's what that stands for. Now, if the colon, especially lower colon, gets irritated, what will happen is it will activate massive amounts of peristalsis. Now, that irritation is inflammation, so many of the functions get shut down, like, for instance, water retention. All right, so if you have inflammation in here, you're no longer retaining the water, and you're also stimulating the smooth muscle, which then leads to watery diarrhea, if you will. And if you have too much diarrhea, you will suffer from dehydration, and that's the problem with cholera. Cholera is fundamentally a pathological irritation caused by a type of bacteria, and it leads to the bacteria release a cholera toxin, and that cholera toxin causes massive inflammation in the colon, and literally you would call it biblical diarrhea. And the treatment is fluid support. We also talk about norovirus. Norovirus does a similar type of a situation, not as dangerous as cholera, but nevertheless significant loss of fluid. So fluid support is quite important. And there is a dedicated defecation reflex. Even though you can withhold it, the anal sphincter is under voluntary control. And so you can resist defecation. You know, young children normally do not have that ability early on, and they have to learn it. But what you get is you get distension because of the presence of mass. That can be solid mass or it can be liquid mass if it's diarrhea. You get stretch. That stretch is then perceived by mechanoreceptors that feed up into the dorsal root of the lower spinal nerve. Then it gets integrated to the pelvic nerve, which is a parasympathetic nerve, to cause not only increased peristaltic contractions, but also triggering a general relaxation of the sphincters. So if you're in the proper place in the right time and you're not incapacitated, what will happen is that you will undergo a normal defecation reflex. You can resist it with voluntary contraction. It can be quite uncomfortable for a minute or two, but after a few minutes, hopefully that first reflex pattern will subside until you can get into a proper location to do your business, if you will. A little pathophysiology here, and I think while this looks quite unpleasant, this is removed from a patient who passed away from a disease known as Hirschsprung's disease. And Hirschsprung's disease is a syndrome. It's an anatomical disorder where you do not have enough innervation of the large intestine. A very, very famous person you might know of suffered from Hirschsprung's disease. His name was Elvis. Elvis Presley suffered from Hirschsprung's disease, and his chronic use of opioids. That's the other big thing. People who have Hirschsprung's or Hirschsprung-like syndrome will often have large amounts of pain and are thus susceptible to opioid addiction in order to manage that colorectal pain. Elvis actually did die on the toilet, not to make too fine a point of it, but he did suffer from. They found out in post-mortem that he actually did suffer from Hirschsprung disease. And so now we're into the last part of the gastrointestinal journey and talking about something that is not something we normally like to talk about in casual conversation. And this is my time to remind you that if you don't have the courage to talk to people about these topics, they won't talk to you about it. Dignity and privacy are given. People want to have dignity and they want to have privacy. So if you can give them the dignity and you can ensure their privacy, then they will open up to you about topics which are normally taboo. In fact, I think it drives a lot of what we talk about in our modern society. That especially, and again, I'm not trying to take a chauvinistic point on this, but we do realize that women in our Western society tend to be very, very reticent to talk about this aspect of their lives, and unfortunately, you probably should. It doesn't seem very sexy. It seems opposite of sexy. But nevertheless, it's extremely important to health. And so I want to remind you to give your patients the dignity and the privacy to allow them to talk about these things. Now, this slide goes under the nice to know. And I understand if you're going to want to bypass it, but let's talk about it for a second. This is a liver metabolic process where you're breaking down alcohol and processing alcohol, ethanol. Alcohol dehydrogenase is the primary enzyme that does this. Now, if you binge drink, you can rapidly swamp this process. Or in familial alcoholism, and this is true in certain minority populations, particularly certain types of American Indian and Asian populations. They have a lower activity form of alcohol dehydrogenase. So the half-life of ethanol goes way, way up. So they have an exposure to ethanol that exceeds what a person with a standard alcohol dehydrogenase would experience. And when you smell someone's alcohol in someone's breath, you're not only smelling ethanol, you're actually smelling this other compound called acetaldehyde. That's actually probably more of what you're smelling. Now, acetaldehyde will also be converted back into acetyl-CoA under low and very, very light drinking. But if you take more than a couple of beverages in any given setting, you're going to go into a high alcohol consumption, which leads to swamping of the system, and it can actually trigger inflammation and damage. By the way, hangovers are primarily due to acetaldehyde. You get dehydrated, and acetaldehyde is causing that sort of wooziness that you feel in a hangover. And this final slide we have just talks a little bit about the idea of what happens when you don't eat versus when you have a, shall we say, a normal scheduled diet. Somewhere around 16, 24 hours, it could depend on your relative amount of physical activity. The glycogen that would be stored in your liver, absent of any other food, will generally deplete to a point where you no longer have a resource there. In those situations, what will happen is the liver will try to synthesize glucose as possible, but that's still not going to probably be sufficient. You'll often feel a little bit fatigued. You'll often feel not as energetic during this time, maybe a little sleepy. But in any case, what's happening is the liver is trying to supplement the glucose that would normally have come from glycogen or a dietary source. So we're going to do gluconeogenesis, that is the creation of glucose. We're also going to oxidize fatty acids that can be found in the liver or can be mobilized from the fat tissue. And when we beta oxidize fatty acids, we are making acetyl-CoA. We're not making glucose. So we're making gluconeogenesis generally from amino acids, and then we are beta oxidizing fatty acids to make acetyl-CoA. Now, if we have poor ratios of insulin and glucagon, like for instance, if we have low insulin and high glucagon, that's going to stimulate lipolysis. That is to say, we're going to break down fats in the adipose tissue, releasing fatty acids, and then we are going to digest those fatty acids and release triglycerides in order to engage in beta oxidation. Some fatty acids are relatively insoluble, especially the saturated fatty acids. They're very, very lipophilic, and so we transport them. You remember how I said albumin is an important carrier protein? Well, here we have a very fatty hydrophobic molecule. Well, that's going to move through the serum albumin. Most of the issue in this step is talking about the fact that once you make acetyl-CoA, you have to transport it inside of the mitochondria, and that's the slow step in this process. All right, and we're making acetyl-CoA in beta oxidation as well as some other molecules again to synthesize ATP. Okay, so there you have it. Getting a little bit dense there on the end. Sorry about that. Again, try to simplify, simplify, simplify. If you can't remember it all, then try to remember short little words, little trigger words to help remind you. You can't hold on to it. You might as well try to hold on to a huge glob of sand in your hand. You're squeezing it tight, and just the sand slips through your fingers. So the key to managing the more dense slides, the more dense material, is find ways to simplify it. Find ways to abbreviate it that you can recall from one or two key words. That's it for now. Thanks for your time, and I will see you when we have class next time. Take care.